The effect of antifungal treatment and oxidative stress in Saccharomyces cerevisiae. During respiration, oxygen is reduced through a series of reactions resulting in the production of reactive oxygen species, including superoxide, peroxides, and hydroxyl radicals. In small, controlled doses, reactive oxygen species can be important in many cell functions. In high concentrations, reactive oxygen species can break down and destroy cell membranes, leading to leakage or cell death. Saccharomyces cerevisiae, along with many other eukaryotes, have evolved mechanisms to combat this damage. The SOD1 gene encodes for the enzyme copper zinc oxide dismutase. This enzyme is able to convert superoxide into hydrogen peroxide or oxygen by alternatively adding or removing electrons in a process called dismutation. Fluconazole is a common antifungal drug that targets the biosynthesis of ergosterol. Cytochrome P450 14-alpha demethylase is an enzyme that cleaves a C14 methyl group from lanestrol to form ergosterol. Ergosterol is key to membrane flexibility, strength, and proper functioning of embedded proteins. Fluconazole targets and interrupts this process. This alteration in the membrane synthesis can severely affect permeability, subjecting the cell to osmotic and oxidative stress. Calcofluro white is considered a fluorescent brightener, an industrial chemical that fluoresces following exposure to UV light. However, calcofluro white is also known to possess antifungal activity due to its ability to competitively bind with newly formed chitin. Chitin can be found in fungal cell walls. Its removal from this structure can severely increase membrane permeability, resulting in reduced cell wall integrity and inhibition of fungal growth. Literature has shown that SOD1 knockout mutants exhibit increased chitin deposition to their walls, indicating that SOD1 is essential for the homeostasis of these structures. As SOD1 is downregulated in response to fluconazole, the deposition of chitin is expected to increase as a means to increase the integrity of the cell wall, thereby enhancing survivability to fluconazole. We took this knowledge and applied it to our three hypotheses. Firstly, fluconazole will inhibit growth more effectively in Saccharomyces cerevisiae that does not have the SOD1 gene. Given the way that superoxide dismutase works and the way fluconazole works, we expected that knocking out this gene out of our yeast cells will make them more sensitive to fluconazole treatment. We expect less growth from our knockout strain than from our wild-type strain. Fluconazole and hydrogen peroxide will have a synergistic effect on growth inhibition for both of these strains. Given that our yeast cells don't like either of these compounds, we expect that both of these strains will have much less growth than treatment with only fluconazole or only hydrogen peroxide. We also expect the knockout strain to grow less than the wild type. Fluconazole and calcofluro white will have a synergistic effect on growth inhibition for both of these strains. With our understanding of the way fluconazole and calcofluro work, we expect that they will have an increased impact on cell growth in both of our strains when added together. Once again, we expect the knockout strain to grow less than the wild type. Let's move on to our observed results. Firstly, it's important to understand how Saccharomyces grows without any treatment. As you can see, we are using a wild type strain as well as a mutant that has had the SOD1 gene knocked out. This prevents the mutant strain from making superoxide dismutase. Using this as a baseline, we can now compare the others. We grew Saccharomyces cerevisiae on agar treated with fluconazole at concentrations of 1, 5, and 10 micrograms per milliliter. Fluconazole has minimal impact on the growth of wild type, however the growth of the mutant decreases in proportion to the concentration of fluconazole. Next, we tested how the wild type and mutant reacted to hydrogen peroxide by itself. Treatment with hydrogen peroxide seemed to have a dose-dependent effect, with higher concentrations favoring the growth of the SOD1 deletion mutant over the wild type. Studies have shown that the presence of hydrogen peroxide can stimulate SOD1 to suppress aerobic metabolism, reducing the production of reactive oxygen species and improving the longevity of the cell. This provides a possible explanation for our results, as the absence of SOD1 may actually improve short-term growth under conditions of high hydrogen peroxide concentration. Now we're going to put these two together. We tested two different concentrations of fluconazole, 10 micrograms per milliliter and 16 micrograms per milliliter. As you can see, the higher concentration of fluconazole severely disrupted growth. But what about calcofluro white? We can see that calcofluro white was as effective as the fluconazole on the SOD knockout, however seemed to have minimal effect on the wild type. Adding calcofluro and fluconazole together drastically increased effectiveness compared to the plates with only one antifungal on them. This was the most effective treatment to inhibit our SOD1 knockout strain, however it seemed to have minimal effect on our wild type. What does this tell us? Treatments with fluconazole gave the predicted results even at low concentrations. That is, growth was inhibited or at least altered, indicating that SOD1 assists in the cell's defense to fluconazole treatment. Some research suggests that SOD1 assists in the reduction of aerobic metabolism in the presence of hydrogen peroxide. This reduction in metabolism could slow the fluconazole uptake, thus improving the survivability of the cell. Further testing would be required for us to say this conclusively. When treated with a higher dose of fluconazole and a lower dose of hydrogen peroxide, the wild type grows better, indicating that this behavior may be dosage dependent. The deletion of SOD1 is known to create significant depositions of chitin in the cell wall. We also tested exposure to calcofluro white, an antifungal drug that specifically targets chitin, presumably increasing membrane permeability. We saw minimal growth in the knocked out strain, while the wild type seemed only slightly inhibited. This inhibition was much more pronounced in the calcofluro plus fluconazole treatment than it was in the treatment that only used fluconazole, suggesting synergistic activity between the two, enhanced by SOD1's downregulation. The results supported the hypothesis that the SOD1 deletion mutant had increased susceptibility to both treatment with fluconazole and treatment with fluconazole plus calcofluro compared to a wild-type strain. This confirms that SOD1 functions to maintain cell wall integrity. This data provides areas of further research into altering susceptibility of antifungal treatment. If SOD1 downregulation can be induced in Saccharomyces Cerevisiae prior to treatment of fluconazole, the efficacy of the drug can be improved. The mutant strain's viability also appeared reduced when oxidative stress was applied, confirming that SOD1 protein functions to reduce the impact of reactive oxygen species. Therefore, without this function, the yeast's ability to cope with oxidative stress was reduced. While some interaction between H2O2 and fluconazole was shown, and growth was inhibited compared to our controls, the SOD knockout strain seemed to grow better than the wild type. As mentioned earlier, this seemed to be dosage dependent and further research would need to be done to completely understand this interaction. Ultimately, this investigation has provided insight as to the future of developing novel and improved treatment of fungal infections. Fungal infections are known to be challenging to treat as most antifungals, such as fluconazole, are fungostatic rather than fungicidal. Therefore, antifungals are often reliant on the patient's immune function to clear infection once immune replication is halted. However, many fungal infections arise in immunocompromised individuals who therefore have reduced ability to combat infection. This makes it particularly difficult to clear infection even with an antifungal treatment. Development of novel methods which may increase the effectiveness of antifungal treatment is therefore a valuable area of research as it can increase efficacy of fungal static treatments, allowing immunocompromised patients to better resist infection and allowing dosage to be reduced, thereby reducing risk to patients. Do you want to check out our primary literature? Here are our resources for everything that we talked about.